know that genes are a gene can be defined as the unit of inheritance, but we can also define the gene as a section of the DNA that codes for a particular protein. Okay, so that means that proteins are actually very important in the discussion of issues to do with genetics. Protein synthesis is very important because, of course, the hormones, the enzymes, hemoglobin, these are all very important proteins that are very important in life. Okay, so that means that looking at the process of protein synthesis or biosynthesis becomes very key. Like I said, the cell cycle, protein synthesis is an important process that also has to be controlled or tightly regulated to make sure that errors don't happen. Because if you have errors in the synthesis of important proteins, then you're going to have problems. For example, you not have certain enzymes that do the work that they're supposed to do, and even issues to do with having the right hormones. You know, a lot of disorders are associated with deficiency of certain hormones. So you don't want to have such a situation, and that's why protein synthesis becomes very important. So protein synthesis is about the process of linking up amino acids to form proteins according to the instruction that is obtained in the DNA. So when we usually say that the DNA directs the function of the whole cell, it is partly because of the fact that the DNA can direct what proteins have to be made. But it achieves this role with the help of messenger RNA. Okay, so messenger RNA is the one that gets the information from the DNA and then takes it to the ribosome, where you will have the proteins being synthesized. Okay, so given that proteins are very important, it is very important that this protein synthesis takes place in the right way. Okay, in plants, the story is quite different from that of humans or animals. For plants, for them to get the amino acids that they actually need, they absorb nitrates, and then from the nitrates they can obtain the nitro group and then use substrates from carbohydrates, especially, or to be very precise, the carbohydrate skeleton. For example, the alpha ketoglutarate from Krebs cycle is what is combined with the nitro group to form amino acid. Okay, in the process of forming the amino acid, then you can have transfer of the amino group to different molecules through the process that we call transamination. So this kind of transfer also influences the kind of amino acid that you will have, or protein that you will have. Okay, so in animals, we can obtain amino acids or proteins from the food that we eat, that is from our diet, but also our body can manufacture amino acids, and the amino acids that the body can manufacture are the ones that we term as the non-essential amino acids. And the direction is that RNA gets the information from DNA to make sure that the right protein is made, and that is why we have this kind of setup: DNA, RNA, and then protein. You can get an objective that illustrates that changes this. You can start with RNA, DNA, and then protein. That is definitely wrong. You cannot have that arrangement. You cannot start with the protein, go to DNA, and then come to RNA. That's also be wrong. So the correct sequence is that information is copied from the DNA and is copied by the messenger RNA, and then a protein is made. And that's the right sequence. So this basically means that the genetic information is stored within the DNA for most organisms, by the way. And then the RNA copies what we just call transcription of the 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 DNA gets its template copied or transcribed by the RNA, and then the proteins are now synthesized on the in the different ribosomes that you have in the cytoplasm, and that means that the process of protein synthesis starts within the nucleus, because that is where you get the DNA for eukaryotic cells. So the process of protein synthesis is actually divided into, let us say, two processes. But of course, in this presentation, I've included this. So in some sources, you can find out that actually amino acid activation is not included as one of the stages or steps of protein synthesis. Okay, but we'll see why it is actually very important to include it. So let's start with the transcription. Transcription is when you have the DNA opening up. That opening up of the DNA is what we call unzipping. Okay, so the DNA unzips. What unzips? Because the DNA is made up of two strands that run in antiparallel direction and also running in a helical manner. Okay, so that means that the bonds that join these nitrogenous bases, we call them hydrogen bonds. These bonds have to be broken. Okay, they have to be broken by the helicase enzyme. Okay, and then that will expose one of the strands of the DNA such that complementary messenger RNA is actually formed from the nucleotides that you have within the nucleus. Okay, so let's go into transcription in detail. A part of the DNA will open up. That part that opens up is the one we are calling the cistron. That opening up is what we call unzipping, or you can call it unwinding. It unwinds because the hydrogen bonds between the nitrogenous bases actually are broken using an enzyme called helicase. So that means that you'll have one strand of the DNA acting as a template. It means that it is the part from which we are going to copy from which the messenger RNA is going to copy from. Okay, that DNA has opened up, and then you have one strand going to act as a template, and then now an enzyme called RNA polymerase. Now, what this should suggest to you is that this enzyme is going to build a polymer. Okay, polymerase. It's going to build a polymer. That means a long strand is going to be made. So that long strand is because you're having RNA nucleotides that are going to be joined up or linked up. Okay, such that you have a single strand of messenger RNA that is complementary. The meaning of that complementary is that the base pairing follows base pairing rules. And those base pairing rules are that where you have adenine, for messenger RNA, you are going to have uracil pairing with it. And then for cytosine, you will have guanine. So that's the base pairing rule that you're going to have, these two here. Okay, so that's very important to also note. Okay, so now the messenger RNA has been made. It gets out of the DNA and exits the nucleus through the nuclear pore and moves to the ribosome. The ribosomes are the cell organelles that are responsible for protein synthesis, and we'll look at their role in protein synthesis shortly. the DNA closes up. That closing up is what we are calling rezipping, such that the double helix structure is restored. Now, the second step, which I said can be skipped in some resources, is what we call amino acid activation. Remember, at this stage, you're having the messenger RNA at the ribosome. Now, amino acid activation is when you have amino acids, specific amino acids, attaching to specific transfer RNA molecules. That's very important to mention because the transfer RNA molecules have to be attaching to the right amino acid. Okay, so each transfer RNA has a specific anticodon and binds to unique or specific amino acid. And that combination forms what we call amino acyl transfer RNA complex. This formation of this complex is powered by ATP. So that means that energy is utilized during the process of amino acid activation or during the process of protein synthesis in general. The enzyme is called amino acyl transfer RNA synthetase, is the one responsible for that. That means, therefore, that depending on what is read or what is on the messenger RNA on the ribosome, the specific amino acid has to be activated, okay, and then brought to the messenger RNA on the ribosome for protein synthesis. This process is very important in that it ensures that the right amino acid sequence will be obtained in the protein. Remember, if you have alterations in the amino acid sequence, the protein that you will have can be defective and you have problems with the cell functions. So we move to translation. This happens when the messenger RNA is on the ribosome, okay, in the cytoplasm. Remember, the ribosomes are found in the cytoplasm. So this process can be defined as a process in which the code on the messenger RNA is actually converted into a protein or into a polypeptide chain on the ribosome. So how does it happen? So you will have the first stage where the ribosome binds to the messenger RNA, okay, and then you will have the first amino acid being brought to be assembled, basically. So the first amino acid that has to be brought often is what we call methionine. This has a code of AUG, and this is what we call the start code, or the start codon. This is the start codon, and if you remember in our presentation about the genetic code, where we said we have the start codon and then the stop codon. So for the process of protein synthesis to basically start, this code has to be, this codon has to be the one that you have, okay? So that's what you have here. You have the ribosome with its major, and this is the major subunit and the small subunit, and then this is the messenger RNA, and these are the codes that you have there, up there. So the methionine is being brought since it's the start codon, okay? And then subsequently other transfer RNA molecules will be brought with the activated amino acids, and then you will have the chain building up, and then consequently a polypeptide chain being released. Okay, so the first amino acid has been brought, and then other amino acids have to be brought. That is what we call elongation. Okay, and the process basically the same. You have the activation of each of the amino acid that are actually being brought. Okay, so the second amino acyl transfer RNA binds to the next messenger RNA codon, and then you have the processes continuing, building on the existing amino acid that you have. Okay, so you will have the ribosome moving from one codon moving forward until you reach a point where you have the stop what? The stop codon. Basically saying, please stop elongating the chain that you are making. And that is when translation actually stops. And then you have the polypeptide chain being released. And that polypeptide chain can undergo different processes. One can be folding it such that it is compact, or you can have modifications such as addition of prophetic groups and others that can happen. Okay, so sometimes you have one messenger RNA strand, and then you will have many ribosomes actually attaching to that single messenger RNA strand. The name for that is what you call polyribosomes, and that therefore means it is possible to synthesize a number of polypeptide chains at the same time. That is what it means when you have polyribosomes. Post-translational modifications. This is when, for example, the polypeptide chain has been formed. There are other changes that will happen, and I've talked about some of them. The folding, the extent of folding determines whether you're having the protein under secondary, tertiary, or quaternary protein, okay? And whether you also have other additions of other inorganic groups, or even addition of other organic groups to form different proteins. For example, hemoglobin, the extent of folding and also addition of iron has to happen. Okay, and then addition of the iron, or for example, the iron being the prophetic group, to make sure that the enzyme can the protein can become functional, because some proteins do not work if they don't have these prophetic groups. And then in my introduction, I noted that actually the process of protein synthesis has to be controlled such that you don't produce the wrong amino acids. So just imagine, you don't produce the wrong proteins. Just imagine that you produce the wrong hemoglobin. If you produce the wrong hemoglobin, you have problems with transportation of oxygen and carbon dioxide, and that could be very problematic. So what cells do is that when carrying out protein synthesis, the right amino acid have to be brought to make sure that the right chain is actually made. But inside the nucleus, for eukaryotic cells, you have a checkpoint. That is, after making the strand of messenger RNA, that strand has to be checked for errors and for removal of nitrogenous bases that will not code for any protein. And therefore, the process basically removal of introns, the introns being sections of the messenger RNA that will not code for any amino acid, leaving only the extrons, the sections that will code for the particular amino acid. Okay, so this is quite very important in making sure that you have the right protein being made at the end of the day. Because if you have errors from transcription, then you'll end up with the wrong protein. It's likely that you end up with the wrong protein. So that's why. So in summary, protein synthesis is where you have proteins being synthesized, the right proteins that will be required by the organism. Because proteins are quite very important, from enzymes, hormones, structural proteins, and many others. If you don't have the right proteins, then you are going to have difficulties in maintaining the metabolic processes. So metabolic processes will get you will get challenges with that. And that means that the process of transcription, amino acid activation, and translation have to be exact, such that you end up with